Male gametes are called spermatozoa and are produced in the testes.
Label E shows the acrosome, which contains enzymes which digest the outer layer of the ovum.
Label D shows the nucleus. This contains highly condensed haploid chromosomes.
Label C shows the mitochondria, which provides energy for the movement of the tail.
Label B shows the tail or flagellum, which repels the sperm by its movement.
Label A shows the microtubules, which produce the movement of the tail and keep the sperm in suspension.
Spermatogenesis is simply the formation of spermatozoa. Huge numbers of these spermatozoa are made in primordial germ cells. There are enormous numbers of these primordial germ cells in the testes which produce millions of spermatozoa on a regular basis. Spermatozoa are the male gametes and have a complex method of production. It starts off with primordial germ cells. These germ cells split into two by mitosis. This goes into the primary spermatocyte stage. In this stage, only one cell survives and goes into secondary spermatocyte stage. In this stage, these cells then split again into four, done by meiosis again. The second split via meiosis leaves four small cells called spermatids. These spermatids then go through a phase called spermiogenesis. This process means that the spermatids develop flagellum, allowing better mobility to swim faster. This is a brief view of spermatogenesis.
This is a short presentation on the female sex cell, the ovum.
A is the corona radiata, part of the protective barrier. B is the cytoplasm. This contains the mitochondria and other organelles. This also has many food reserves. C is the nucleolus. D is the nucleus. This holds the genetic information. E is the zona pellucida. This is the protective layer that initiates the acrosome reaction. Female gametes are called ova and are produced in the ovaries. They are the biggest cell in the body, around 0.1 millimeters across. Only thousands of ova are produced and will run out within a lifetime. Each primordial germ cell produces only one egg.
Oogenesis, the production of cells, egg cells in females. It has several stages. First phase or stage is multiplication. You're given a germ cell with a full diploid number, that's 46 chromosomes for humans. This then goes into mitosis, which separates into daughter cells, also with a diploid number of 46. These are called oogena. It then goes into a growth phase where one of the daughter cells, called a primary oocyte, gets ready to separate into meiosis, by meiosis even, and this takes place in the ovary of fetus.
The growth stage begins with a primary oocyte, which is ready to go into meiosis, and in the first stage of meiosis, we begin with a secondary oocyte and a polar body which may or may not divide. This then goes into the second part of meiosis, where we are left with 3 polar bodies which eventually die, and an ovum which is relatively larger than the polar bodies. This ovum will then be ready to be fertilised at a due time. Yeah.
And that's all the stages of oogenesis.
Start it.
Today we'll be talking about the acrosomal reaction. Over to the Lapo.
At 1, we see the sperm has reached the follicle and will later trigger an acrosomal reaction. At 2, we see the sperm has made contact with the zona pellucida, which causes the acrosomes to swell, then binds with the surface membrane of the sperm and releases the vesicle containing hydrolytic enzymes via exocytosis.
Hello and welcome to this quick presentation of the process of fertilisation.
At point A, it shows the sperm reaching the egg cell in the oviduct when it makes contact with the zona pellucida, which is the protective layer around the ovum. This is when the acrosome reaction occurs, which is when the acrosome in the head of the sperm swells and releases digestive enzymes.
At point B, the enzyme digests the zona pellucida so the sperm can move through it to the cell membrane of the egg cell.
At point C, it shows the the point where the sperm head fuses with the cell membrane of the egg cell. This triggers the cortical reaction in which the egg releases the contents of the vesicles called cortical granules into the space between the cell and the zona pellucida.
And finally, at point D, cortical granules makes the zona pellucida thicken, preventing polyspermia, which is when more and more sperm goes into the egg. Only the sperm nucleus enters the egg cell. Tail was discarded. Nucleus of the sperm fuses with the egg nucleus.